We put together a few questions that we thought we could start with. Is that what's on the slide,
Yeah.
Tony?
Yeah. Okay. So we very last thing just before the meeting this morning came up with our first thoughts about some of the sort of themes that have come through from the meeting, which hopefully we can get your thoughts on, and please add any that you would like to discuss as well. So we sort of divided up into a bit of a basic, a clinical, and then a sort of a drug development type program. So if I just run through them quickly and then we can take each one in turn. So the first was, and I think this sort of is existing quite well at the moment, especially in the prader-willi field of collaborations with people with animal models. But I think what came out from this meeting was the potential to collaborate with people, as, for example, Rachel is already doing, with people who are a little bit more expert in some of the aspects of feeding behavior, and particularly around the possibility of testing in animal models drugs and particular types of bariatric surgery, as Randy Seeley mentioned, which may be a good way to get proof of concept before embarking on the difficult human studies. Secondly, what sort of further international collaborations we can set up, not necessarily just Prader-Willi, but obviously with the other syndromes and diseases as well. We haven't really talked much about brain banks at all. As many of you may know, the Netherlands Brain Institute with Dick Swab has a number of Prader-Willi brains. Whether that exists in the other syndromes, I don't know, and it'd be useful to hear from members of the audience whether there are those in existence for Bardet-Biedl, etc., etc. And obviously, it doesn't just mean brain now, with all the interesting stuff we're hearing about epigenetics and the stem cells, the possibility of tissue and cell banks for other things. Then moving on to the more clinical aspects, from Jack's talk and from some talks from Mate, about maybe we really need to try and get some standardized techniques and methods and questionnaires that are usable across all the diseases with Prader-Willi syndrome, and certainly within diseases such as Prader-Willi, but also with the others, so that when we want to do drug trials, we've got things that we can use. And maybe also, as I mentioned, one of my questions about a questionnaire about the food environment in particular would be useful to develop that. And that may need, as I was talking with the people from Ferring this morning over breakfast, collaborations with pharmacy and people like the FDA, so that we've got questionnaires that they will recognize as outcome measures for drug trials. And I think that would be really, really useful if at this meeting we can get that set up. I think from the bariatric surgery point of view, there were some interesting things that came out. One of the things that struck me is the publication bias that there is, and it might be interesting for us to try and set up an international survey through the International Prader-Willi Association, and maybe similarly through the Bardet-Biedl associations, etc., to try and get a feel out there what the true response to bariatric surgery has been. Finally, with things like the stem cells and animal models, what sort of drug screening platforms could be set up, and then also broadening out into the as yet undiagnosed patients, as we heard in the genetic talks yesterday and from Dan from his emo cohort, how we can collaborate to improve those. So that's enough for today's discussion. So should we start at the beginning? Animal studies collaborations and links with people who can test drugs and surgery. Any thoughts about that? And please talk, because otherwise I'll get a sore voice. Someone got microphones?
Let's get the mic around.
That's a good list.
And please, just so everybody knows who's talking, and introduce yourself when you do so.
I'm Teresa Strong from the Foundation for Prader-Willi Research and also from UAB. So I know many in this room have thought about this, and I've thought about it too and haven't come up with a great answer. But what is the animal model for PWS that you use for these obesity drugs? Because there's no perfect model, and if you test it in MAGL2, for example, which gets obese, and it doesn't work, does that mean you wouldn't take it into humans? Or so in thinking about going to the FDA or just proof of principle, what is the model to use?
Rachel? So put you on the spot.
I think we're going to have to wait and see how that pans out, but certainly, like I said yesterday, there are at least 3 of the mouse models that have components of energy imbalance. And so there are measurable quantities for all 3 of those mouse models that could be targeted by drugs in different ways. And so I think if you didn't see something with one particular agent in one mouse model, you'd still have to look at the other mouse models.
And if I can comment on that from the human perspective, I think what Teresa said is exactly right, that just because something does or doesn't work in mice doesn't really tell you very much about what will happen in the human condition. So we are required to consider studies in humans, even if things don't work in mice. You can't be sure. But I think that most drug companies are not going to want to move forward with large clinical trials unless they have some model. So I think that's got to be a major impetus to continue to develop the mouse models and maybe even extend to other organisms. And that, of course, is much harder to do. But there may be other organisms that will serve as a better model for prader-willi syndrome in terms of what happens when you affect this imprinting area.
Christian, Val, or Andrew around can comment about where they feel in Bardet beetle, craniopharyngioma, and SIM1, the sort of screening for treatments, what your strategies are in those models.
I'd like maybe Rob to comment on his pig models.
Well, what I can say is that right now we don't yet have a pig model. We're working on that. Because we don't have the severe hyperphagia and obesity in the mouse models, I decided to try to work on a different animal model, and we've done the genomics so far, but that's where we're at. So pig, perhaps other animal models might be suitable, but we need to develop those before we can really assess both the pathophysiology, do they develop the hyperphagia and obesity, and then think about therapeutic approaches. But we're working on it.
Hi, yeah, I'm Tom Hughes from Zafgen in Cambridge. So we're sort of faced with the opposite situation. We have a compound that might have real utility in a number of different settings of hyperphagia, and we do have human data as well showing that we see reduction in body weight and reduction in appetite as well, or hunger, I should say. And so we're very, very open to working with groups to explore the action of the compound to understand better the mechanism of action. And I think, based on conversations we had last night, even a negative result in an animal model could be quite instructive to us to teach us where things aren't working. I mean, you've heard over the last few days knockouts where you look for lack of action or loss of activity. And I think either way we learn something from it. We're a tiny company, though. We don't have the ability to set up our own models or even to test them. And so we just welcome input or ideas from people, or if there's a way to test our compounds in your setting, we're happy to do that. And we have prototype compounds as well that don't get in the way of our intellectual property or anything like that. So we're open to collaboration, and please do reach out to us. You can find us somehow, or just find me or Jim Vath here while we're at the meeting. Thank you.
Could you say the company again? I'm not sure we name the company.
Zafgen.
Zafgen. So
ZAFGEN.com.
Yes.
Christian Roth, Seattle. I presented yesterday our rodent model of hypothermic obesity, and this rodent model mimics more the hypothermic obesity in patients with lesions, and I think it would be difficult to mimic that in a mouse model because of the type of how we do these lesions, but in rats it works pretty well, and we find the main features of patients with suffering from hypothalamic obesity in these rats. So we could test effects on hyperphagia, but also on energy expenditure.
And I think the same is probably true for some of the Bardet beetle syndrome mice. Is Dr. Sheffield here? Okay, well, my impression anyway of review of the literature is that the mouse models look pretty good. And maybe Rudy, you want to comment? They look pretty good in terms of both the eye phenotype, the obesity phenotype, and even the skeletal, the polydactyly phenotype. So, I mean, I think we have some mouse models that will be very useful for testing treatment strategies, but others, unfortunately, not quite as convincing. But regardless, they'll all have to go to human trials, early trials, and that means taking patients who are certainly often not able to make decisions by themselves and asking them to participate in clinical trials.
Christian, could I ask you how systematic and what's your way of thinking to take it forward? I mean, you've got a good animal model. How are you going to decide what to test and therefore what to take forward, hopefully eventually into a human trial? Is there a sort of a progressive plan to approach that?
Yeah, so we started with some drugs that would make sense from a physiological standpoint. I would like to focus on first single drugs and then combinations of two drugs, because that would make sense. We delete several weight regulating systems that are redundant, and I think that's a key difference between our model and knockout model, for instance. And therefore, I think the value of our model is that we have this complex interactions and complex lesions. So one would be melanocortin signaling, but then also hindbrain signaling, exendine, for instance, would be something, or another GLP-1 agonist.
Hi, I'm Jim Travascus from Novartis in Cambridge, Massachusetts, and we're a little bit bigger than Zafgen, and we probably have, we've got the ability to do those large trials, but the model that we use is more of a proof of concept approach, where you would take your drug target into a population that theoretically would be hyper-responsive or give you the best result that you could get. So in this sort of example for obesity or hyperphagia, finding a target or a therapy that would be selective or perhaps beneficial mostly in prader-willi is a great flagship approach for what Novartis would like to do. And then theoretically you would get registration for that compound that way in a small population or smaller population, but it still would hopefully translate to obesity on a larger scale. But it all comes back to, I think, predicting how your target's going to work. It's going to be target dependent and individual, right? So I think you just have to, keep learning about the biology. I've been impressed with the approaches, but I think, what may work for Prader-Willi but might fail for general obesity, I don't care about that. If it works for Prader-Willi, that's fantastic, and Novartis would look at that.
You want to go on to another question?
Sure. Yeah.
Yep.
Consortia, I mean, apart from the ability to travel around the world and meet everyone, what's our benefit of that? And obviously with the Prader-Willi community, we have reasonable meetings and fairly regular events internationally. Is there some way of is there a benefit in more formalizing the links between the different syndromes and diseases, and how would we go about that?
I assume you're talking about a hyperphagia consortium?
Yeah.
There are already some consortiums in the U.S., right? Is that right, Dan?
Well, there's a rare disease consortium.
Rare disease.
With Heymsfield, Rett, Prader-Willi.
Mm-hmm.
I see one of the challenges, it's something we kind of were looking at before, is that, I'm the Vice President for the International Prader-Willi Syndrome Association, IPSO, and we were looking, Georgia and I were looking at and calling and contacting other rare disorders, but most of the other rare disorders are not very organized groups, the hyperphagic ones I'm talking about, and that was a problem in pulling that together. We're not in a position to put all the effort on it. What are your thoughts, the panel's thoughts on how that could happen? And then Linda.
Well, I guess it's also at two levels. I mean, there's obviously at the parent, carer organized level, and you guys do a great job, which none of us scientists have the time to do in getting these people, everyone together. And I guess there's also at the science level, slightly removed maybe from the involvement of the parents and patients. I mean, Val's gone, but I think the Bardet Beetle community has worked very well internationally collaborating. Is that my understanding, that they sort of, do they parcelate up the 17 genes between different groups and things like that?
Well, Alstrom has an international group too, yeah.
So I guess the question is whether outside of these sort of meetings, which have been fantastic, whether there is an extra benefit in bringing together the various diseases. I mean, what's been interesting is how actually they're not as separate as we may have thought, in that there does seem to potentially be, in the POMC neurons at least, a common mechanism of action for many of them. So actually we may we may have a reason to come together more scientifically as well as culturally and educationally.
I think there are additional potential advantages to international organization, which would go beyond meeting, but also for exchanging research materials and potentially maintaining tissue and cell banks. If the kind of funding is available to support that, it could be extremely helpful for investigators as they try to answer different questions. And even when we try to organize very small collaborations, sometimes samples are hard to come by, surprisingly hard to come by, because of limitations in the way they were collected, or the proprietary nature of who holds samples that could be overcome by an umbrella organization. And I think we could make potentially a lot more progress in hyperphagia if we were willing to support that kind of organization, and only perhaps supplying funding to folks who would then supply samples back.
Bigger isn't necessarily better, but one huge thing that is getting organized is Obesity Week in November of 2013, which just sort of fortuitously, it's in Atlanta and it immediately follows our prader-willi national meeting. But the Obesity Society, the ASMBS, which are both big organizations, are behind that, and they have a whole consortium of a number of other organizations. I am assuming that if that's very successful, they'll start to make that an annual or biannual event.
That's planned now for the next 5 years to be a co-meeting. I think all of us are members of the Obesity Society, and I think, again, it's a matter of funding whether hyperphagia specifically could be part of that conversation. But at a minimum, we might think about sponsoring a symposium, or at least supplying a symposium for that joint conference, so as to continue the exciting directions that have been proposed here and will continue to be proposed.
Yeah, that's a good idea.
I mean, I can't remember where we used to have a prader-willi section in the Obesity Society, which sort of lapsed a bit. And maybe we can use this as a way of resurrecting a hyperphagia section of the Obesity Society.
Yeah, I know the
Jack,
leadership is
you're pretty involved with the Obesity Society, right?
Yeah,
All of you. Okay.
Yeah, I think they'd be open to considering that. Again, it really is a matter of making sure that we continue to have an additional purpose beyond what's done there. So I think we have to make sure that we are well represented at those meetings if we're going to be a presence.
Any more comments on that, or do you want to move on to the next?
Tissue banks. So, I mean, I have a bit of experience from the Netherlands Brain Institute, where I did some work as a PhD student on the brain bank side of things. There are some logistic problems, as Dan will attest, to getting brains to Amsterdam. But maybe people want to comment, maybe Rudy, about cell lines and how we might go about getting some sort of international collection together.
Yeah.
Yeah, sorry.
For reasons that hopefully at least came across partly in my presentation, I think that's a very important step to take. We've started to do this with support from the Helmsley Foundation Leona Helmsley and the dog. You all. So that finally got settled, and some money got freed up. And we've built a bank so far of about 130 cell lines, half of which are now in IPS status from individuals with varying kinds of diabetes, including type 1 and type 2, and various forms of monogenic obesity. And at least based on the sort of preliminary evidence that you heard today, I think we're going to be able to model aspects of disorders like Prader-Willi and Bardet-Biedl and other complex types of obesity, and maybe even ultimately some of the more common types, by using iPS cells, differentiating them to different kinds of neurons ultimately, and then using those to interrogate basic biological mechanism, but also for high throughput screens of one kind or another. So small molecule screens, it's pretty easy to imagine. Once we've got cells that we have confidence are fully recapitulating the phenotype that would be anticipated from the individual, can be subjected to high throughput screening with, I think, a high likelihood of finding some very interesting interventions and also discovering other aspects of the disease. So I'm all for it. And the way that we've done this locally is we have IRB approval to take small skin biopsies. As a matter of fact, some of the physicians, when we started this, the physicians were not comfortable working in the diabetes center doing skin biopsies, but when we found out that the dermatologists were going to charge us $400 a biopsy, suddenly a lot of people realized they could do this. And in fact, it's the case. It's a very simple procedure. It's essentially painless, and the transporting of the tissue is really quite straightforward. We've dealt with Dan Driscoll on this point as well. So I think it's a very, very good idea. We could set up a central repository for these things. The critical point, I think, is to have the cells banked. They don't necessarily have to be made into stem cells immediately. Some of the phenotyping can be done on the fibroblasts, as I showed you. The fibroblasts show some of the anticipated phenotypes. Ultimately, obviously, the brain will be better. And the most important thing, or certainly equally important, is very sophisticated phenotyping. So, and genotyping, at least in case of to the extent that's available for the Prader-Willis and the Bardet-Biedls and other similar disorders, because obviously that will inform some of the kinds of experiments that individual labs may want to do, and particularly these microdeleted Prader-Willi patients, as we've been talking about, I think would be enormously interesting from this point of view, as well as reagents that others have developed for manipulating genes that are in the, for example, the Prader-Willi locus or some of the Bardet-Biedl genes. These all, to the extent that these are more widely available, I think it will expedite this work. And the people in this room are obviously much more expert in terms of some of the biology and clinical aspects of these disorders, and your insights, I think, are absolutely critical. If you see patients that you think may be informative in one way or another, these would be ideal to add to a bank like this, even if you don't have a specific diagnosis yet.
Could you just comment, Rudy, on I remember seeing a very interesting documentary about Henrietta Lacks, who made the HeLa cells that then went around the world, and there was a bit of a negative comeback about that. Could you just comment on the sort of ethical and intellectual property issues around these sort of things, and if those are something we can just forget about, or if there are implications?
So it depends on who you're dealing with, the extent to which the, quote, intellectual property issue arises. In our consent form for the skin biopsy, what we indicate to the patient or ask the patient to indicate is, A, can we do any experiments that we wish, or are there any that they want to exclude? And secondly, can we distribute the cells to others ad libitum? And I would say to date, 100% of our patients have signed up for both. And the issue of intellectual property, we've not tried to recover any of that in the cells that we've distributed. And I think one of the points of agreement ought to be that anything that gets into a bank like this really is the property of the community, with some careful vetting process for letting the cells out. I don't think this should be something where you can mail in a vial of, of tissue culture medium and have somebody send you number 28, 35, and 56 from the bank. There ought to be some vetting process for what the individual proposes to do with it and why. I mean, not obviously in enormous detail, so that they don't end up in promiscuous sort of use. But and the agreement going in ought to be that that sort of vetting process will be set up so that it avoids misuse of these.
Junan?
So I work at the UCI with the Rare Disease Consortium, and all of our patients now, if they want to, can be consented, and their cells get sent to Coriell. So they consent to the process, and only researchers who put into Coriell, I believe, can take out of Coriell. So they're increasing. When we first went to them, we wanted some cell lines from them of prader-willi, and they only had 2 individuals. So that prompted us to start doing that.
And how is that supported? How is that paid for?
It doesn't cost us anything to send them, so I don't know how it's supported.
Yeah, well, that is, you mentioned that, Jack. I deal with the deaths at Prader-Willi, and we've got 2 issues there. One, we tried to work with Dick Schwab on the internationally on the brains, because he's our biggest brain expert. But the cost of who's paying for what in getting the brains overseas was the issue there. So we never did get that quite resolved, right, Tony?
Yep.
And Dan? So the other issue we've got is, of course, parents consenting ahead of time, and if they don't, then there's the death, and I often find the medical examiner discourages an autopsy or anything else, and by the time we get the news of the death, it's unless I get a call that they're dying, it's often too late, and they've often been discouraged, and then there is no autopsy in that. So that's an issue. But somebody gave me an idea here about, and we certainly encourage brain and tissue donations. But I think maybe at our next national conference that involves a lot of parents in 2013, we can actually have the brain and tissue bank come, and I guess they can sign up at the conference, families. So I think that might be since we get a large audience, that might be a good way to encourage people to sign up ahead of time and avoid that last minute trauma. Or even if they read it in the newsletter and on the website, or we encourage them, they think about it. I have to admit, I haven't even signed my own son up, so, and I know better. But you think about it, you never quite get to it. So if we have somebody there, I think that might be a good way to look to potentially do it and get people signed up, as long as somebody's there that can sign them up on site.
Yes.
Another idea would be to bring it up at the providers meeting, to discuss it with them, and to have like a card that they could give to parents when they interact with parents at the yearly meeting.
That's a good idea. Yeah, to work with the providers more on it, yeah.
I think it's very important that the society consider creating an advanced directives kind of model forms that patients could, or parents and patients could fill out together and decide what they would like to have happen. Mm-hmm. It's a really useful exercise. It's not a pleasant one, but it's a very useful exercise and can really help take away a lot of the stress later on. But I think it's important to develop the model language for people.
Maybe I'm jumping ahead, but Rudy mentioned about the phenotyping. Rudy, is there anything like a core phenotyping that you would do? It's going to be so variable, based on individual patients.
So for some patients, obviously, we can come back to that, but for Prader-Willi patients and Bardet-Biedl and Alstrom and others of these monogenic forms of obesity, people who are encountering these patients will know more about their subphenotypes than I do. That is, the experts in this room would be able to tell you what might be the difference between a microdeleted Prader-Willi and an individual with a larger deletion. And I think the more sophisticated the input with regard to those phenotypes, the better the use of the bank will be. And it's hard to specify. I mean, what might be relevant for Bardet-Biedl won't necessarily be relevant for Prader-Willi or Alstrom, but for each of these, the more sophisticated the phenotype, the better, with regard to, well, you guys will know better than I do. For patients who have obesity that is not clearly in any category, the phenotypes that one would want are, again, the phenotypes that many of you are very familiar with. What was the age of onset? What is the characteristics in terms of the growth of the child, in terms of body weight, height, length, so forth? What characteristics in terms of feeding behavior that might be unusual or particularly characteristic? Just, I think, the sort of sophistication that's in this room would be, if it were necessary, you could obviously make a form up, but my sense is that even that wouldn't be necessary, just to simply have very careful history taking, very careful family history, and in instances in which there's a specific genetic diagnosis, many in this room will know the subtleties, for example, of Bardet-Biedl 1 versus 10, that sort of thing. It would be very helpful to have as much phenotypic information as possible.
So, I mean, apropos that, I have to say that from my experience of trying to do detailed phenotyping, it really helps a lot to have a standardized form with checklists and boxes with common features that can be filled out. And again, that would be something that the society might want to work on, is a standardized set of questions to be answered by providers. Now, it's not ideal because it's not being assessed by the same individual, the same team, but there's certainly a lot of advantage to having a standard question answered in a standard way. Dr. Joan Hannen has been working hard on Wagner syndrome, doing a comprehensive phenotyping protocol and receiving voluminous medical records from each patient. Extracting those is an arduous task and is not always very satisfying. But the comprehensive phenotyping that can be decided on in advance makes a huge difference. And I don't know if you want to make a comment, Sean.
Yeah, you can imagine setting up a set of forms, if that was what was desired, that would be key to specific suspected or actually confirmed diagnoses. That would make a lot of sense. So for one diagnosis, it might be more relevant to record one thing or another. And then in circumstances in which the diagnosis is unclear, other than that there's severe obesity, then the kinds of questions might be different.
But you might not want to skip those questions because of the unexpected findings that appear. That's certainly been the case in Wagner syndrome, that we've discovered certain aspects that we didn't know about in advance.
Do you know anosmia was not appreciated in Bardet-Biedl for many years because nobody bothered to ask the question. So I'm sure there are other examples.
And certainly, the tissue banks are obviously a great idea. Ideally, practically, they're expensive. And, to echo what Rudy and Jack have been talking about, getting precise phenotyping and genotyping is crucial. And there was a NIH brain bank that had Prader-Willi brains, and I've had 2 instances where I know that those weren't Prader-Willi brains, that they said were Prader-Willi brains. And when we genetically looked at it, one, well, I won't mention names and investigators, but I think that I think it's really important to have well-funded banks, because if you don't, and long-term funding, you're not. It's going to be garbage in, garbage out, and this brain bank I'm thinking about was an NIH-funded brain bank, and like I said, 2 brains, supposedly Prader-Willi or not Prader-Willi, genetically, and the clinical information is very sparse and sketchy anyway.
You know, you had talked about having a more exact phenotype. One of the problems we found out with prader-willi is we got so exact, they started ruling out kids, you know. Oh, no, their IQ's too high, they're this, they're that. So we had to reduce, you know, the phenotype we were posting because they were ruling out kids and not examining them for prader-willi, you know, doing the testing on them because it was too exclusive. So that's the other extreme, you know, the danger there.
Speaking on behalf of the patient community, I think that's one of the places where we feel like we can perhaps be contributing as far as long-term funding for a tissue bank. I mean, ideally, we'd like to have PWSA, of course, has an electronic database. We're working to expand that as part of the Global Rare Disease Registry project at the Office of Rare Disease at the NIH. And ideally, we'd like to eventually link that to a tissue bank so we would have the electronic information linked to either, and that's, I guess, the discussion that needs to happen. Would it be iPS cells? Would it be PBMCs or fibroblasts? And I think that from the patient advocate standpoint is a great place for us to support and move that along. So we'd be happy to be involved in that.
That's great.
Okay, so next is standard evaluation protocols to help with clinical assessments and drug trials. I'm going to put people up on the spot, but maybe Mate wants to comment, and maybe someone from Ferring could comment as well. Mate?
Yes, the point was not to standardize evaluation, but to make a comprehensive evaluation. I think it's very difficult to try to have standardized evaluation when evaluating a drug, you know, because it depends on the way and depends on the environment where we are doing the trial. So, but at least when we use a drug for hyperphagia or eating behavior, to have the complete evaluation of behavior, general behavior of the food environment and on the eating behavior. That was I wanted to say.
Merlin?
Could I back up one step back to tissue banks? There are several of us in the audience that are, well, I guess we all are. Telomeres are getting shorter. We're getting older. And we've accumulated samples over 3 decades, hundreds of cell lines from patients, plasma samples, serum samples. What happens with those cell lines once the PIs retire? Has that been considered? I mean, that's a... Dan has hundreds of cell lines. I've got hundreds of cell lines. There are probably others in the audience here. You know, we've been involved with prader-willi for 32 years. And we've seen patients that length of time that's accumulated thousands. I've got thousands of plasma samples, tens of thousands probably. If you start looking at all the different samples we accumulate over time, I mean, it adds up. When you over 32 years, it's because we're not good at it, just because we've been around a long time. But what happens to this? I mean, is that something? What are other rare disease organizations doing with these types of situations? Is that a problem we have in the States? I'm sure it's a problem in the UK and in France. And
it's a general problem.
My problem
succession planning.
My problem in the UK was solved because my freezer broke and I lost all my plasma samples.
That's why you eliminate some samples. Yeah. So what happens? Does anyone have any words of wisdom or advice?
Steve?
No?
I don't talk about succession planning.
Merlin, as you know, it's a sticky issue too with the university. I mean, you had, if you don't mind my saying, I mean, when you switched from Children's Mercy Hospital to Kansas University Medical Center, CMH was saying those were the CMH's samples and gave you a lot of trouble to even move it down the street to another institution. So that's another obstacle is the academic or private institutions saying, we own the samples, not the investigator. And actually, you know, all of us that get funded by various funding agencies, mine goes to the University of Florida.
And the patent issues come up too.
Right.
We've only got a couple of minutes left. Can I just come up with a couple of things? Firstly, what do the audience feel about setting up a working party consortium amongst selected, just to keep the numbers down, party to come up with a standardized protocols for different ages for the investigation of Prader-Willi syndrome hyperphagia for use in both phenotypic assessment and drug trials, which then could be internationally used. So we'd have representations from everyone, maybe have someone from the drug industry involved, maybe someone from the FDA involved. I think that would be a tremendous outcome.
If you agree, to raise your hand.
Okay, good idea.
Okay.
Who's in charge, Tony?
To be debated. Okay. I think we may have to, I mean, the bariatric surgery, I've already chatted with Jan Lee that we can do. Links with industry and FDA we sort of mentioned. And I think we're probably out of time to discuss the rest, unless anyone has any burning questions that have been unsatisfactorily answered.
We could go a couple minutes over it. Do you want to mention the questionnaires, since that was a big issue brought up?
Yeah, I mean, I guess part of that was who would like to help develop a questionnaire for the food environment? Okay. So we've got a few there. So that's Barbara, Linda, Janice, and someone else.
There are also some investigators within the psychological
Yeah,
study
well,
of
great.
eating that would be very relevant for that kind of task force.
Greg Chirpy's in back also, said he wanted to be on it. And Julian wanted to be on it. Yeah, Barb, they got you.
I think it's lunchtime.